struggle with free energy? What those concepts are? So this is like an unfavorable reaction, right? Adding that phosphate spontaneously to glucose to make glucose 6-phosphate. But of course, this is spontaneous. Get a lot of free energy, negative, free energy release from taking ATP and hydrolyzing a phosphate from that to make ADP. And the way we do this in the body, to get this phosphate added to glucose, which is the first step in glycolysis, this is catalyzed either by glucokinase, and that's actually for storage of sugar as like glycogen, or hexokinase is the enzyme that really enters into glycolysis. Is this phosphate gets hydrolyzed basically by the glucose. So you basically sum those two together and you get negative free energy. It's just not as much negative free energy as you would have if it was just, hydrolyzing ATP phosphate. It's basically this minus this amount, which gives you this. It's kind of hard to tell because they're all on different levels. Right? Anyways, so this free energy of activation, what is that? Why do we have to do that? You know what this represents here? Like the top of that peak? Transition state, right? So it's basically energy you got to put into it to break those bonds and form the new ones. So this represents your transition state. And how do we do that with catalysis? How do we lower that energy of activation with catalysis? Basically you stabilize that transition state. And I think we'll talk about that more later. But catalysis is, a catalyst is a compound that increases the rate of a chemical reaction. And they lower the activation energy of free energy. They lower the activation free energy, or the energy of activation, basically. Does not alter the standard state free energy change of that reaction. It just lowers the amount of energy you have to get to that to that transition state, or basically energetically stabilizes the transition state. That's one characteristic about transition states, is they're so unstable that they're not going to last very long. But catalysts, you find, especially enzyme catalysts, are built to favor binding the transition state rather than either the substrate or the product. And in that way, in fact, they favor binding the transition state. They stabilize that transition state, lowering the free energy of that transition state, which lowers the free energy of activation. And it accelerates the reaction. Enzymatic catalysis accelerates it under mild conditions. It's also highly specific, so it's only going to accelerate the one reaction you want. When you guys took organic, when you're doing like synthesis, like what would be your yield typically for like one reaction? Might be lucky at like 40% or something like that. But enzymes typically have high specificity, so it's going to favor one reaction rather than side reactions. You typically get yields, in the upper 90% range. And then enzymes give you the possibility for radiation. You want to be able to shut down pathways when you have, plenty of the product, plenty of energy. You don't need to make ATP. It's time to store energy as glucose. And likewise, if you have a low amount of energy in the cell, you need to start using glucose to make ATP. So you need to be able to regulate these pathways. And I think I drew this picture on the board last time, right? This just shows you how you can increase the rate of reaction by increasing the average free energy of the reactants, and it does that by just bringing the reactants closer to that energy of the transition state. And that's how it accelerates those reactions. It lowers the free energy of activation because it raises the energy of the reactants. Whereas enzymes themselves lower that free energy of the transition state. by stabilizing that transition state. And that lowers the free energy of activation, increasing those rates. This is the same picture as that second one, basically. I always like this little animation, just showing you in just blocks what the free energy of activation is. First, you have to put energy. You have oxygen and sugar here. Put energy into it to break all the covalent bonds, and then you get a nice yield when you recombine all those bonds, oxidizing the carbon and hydrogen to make carbon dioxide and water. And this is what we'll see a lot, molecular pathways in biochemistry, where you have some entry into the pathway that's going to yield this final product, and there's several steps in between that are all enzymatically catalyzed, and typically this first enzyme is going to regulate the entry of substrate into that pathway. And you get, a lot of times you get feedback inhibition, where the accumulation of product F actually will inhibit or slow down, the activity of enzyme 1 that will stop feeding A, substrate A into that pathway. We'll see this a lot in metabolic pathways and signal transduction pathways. Do you guys have any idea what signal transduction is? Somewhat, nerves do signal transduction, but almost every cell in your body does also. So let's say insulin. What's insulin? It's a hormone to tell your cells to take glucose up, right? So how does it do that? It binds to a receptor on the outside of the cell. Once it binds to that receptor, that triggers a signal pathway within that cell to tell the cell to start fusing glucose transporters that are vesicles, not in the membrane, but just inside the cytosol. Those vesicles, with the signal transduction pathway from the binding event at that receptor, signals to the cell that those vesicles fuse with the membrane, and you end up with those glucose transporters there on the outside of the cell that can then take up the glucose. So the signal transduction pathway is that pathway that goes from the signal, the insulin binding the receptor, all the way to the nucleus. You start making new proteins, and also triggers kinases and phosphatases that will get those exosomes to fuse with the plasma membrane. So you guys have no idea what a kinase is, or a phosphatase. So these are probably the two biggest enzymes that you'll see in biochemistry involved with signal transduction. Kinase is just and glycolysis, metabolism. Kinase is just an enzyme that adds a phosphate group to something. A lot of times it's adding a phosphate group to a protein that alters that new protein's function. Phosphatases just do the exact opposite. They hydrolyze phosphate groups off of proteins or other compounds. So, yes?
Is that used for ATP? Is that used for ATP, like the ATP becoming ATP?
Yeah, a lot of kinases use ATP. There's a couple kinases that make ATP. Yeah, so pyruvate kinase actually makes ATP. It takes phosphoenolpyruvate, which is the enol form of pyruvate. Now the keto-enol shifts we talked about last time a little bit. So phosphoenolpyruvate is a high energy phosphate compound that has the phosphate, and pyruvate kinase. Basically, that reaction runs in reverse of what you would normally see at kinase. So that the phosphate from the pyruvate gets added to ADP to make ATP. And that's a form of phosphorus. It's called substrate level phosphorylation. There's only a couple of reactions that make ATP that way. But they always have to come from a higher energy phosphate down to be able to form that medium energy phosphate, which ATP is. So you'll see intermediates that can do that are phosphoenolpyruvate and dihydroxyaldehyde, glyceraldehyde. glyceraldehyde diphosphate also. So pathways that are controlled, they're regulated by the metabolites. This is just what I just showed you. And this is an example where they actually use real compounds. Threonine's the entry pathway to produce isoleucine. You guys know what threonine or isoleucine are? I don't expect you to yet, because amino acids? Yeah, they're amino acids. Right? So this is the synthetic pathway for isoleucine. Threonine is the entry point for that. And when you have a lot of threonine around, that'll come back and block the enzyme 1 activity into that pathway. So this is an example of negative regulation. Product of enzyme 5 inhibits enzyme 1, because if you have plenty of isoleucine, there's no reason to keep using threonine to make it. You could probably use it for other things, like building proteins or making glucose burn for energy. So genetic and evolutionary foundations, life on Earth, we think arose about 3.5 to 3.8 billion years ago. And the formation of self-replicating molecules is a key step. Self-replicating molecules. Could it have been DNA or proteins? You guys know? They tend to think it was RNA was the self-replicating proteins. Have you guys ever heard of this experiment? Was it the Miller-Urey? Yeah, Miller-Urey experiment, where they put a lot of compounds in there with what they think the primordial soup of the earth might have been, had its spark, and then they were able to eventually produce hydrogen cyanide plus amino acids, simplest amino acid like glycine. And since they've done this, I think they discovered actually glycine on comets and stuff. You guys heard of that? Yeah, so maybe this is all a farce, but you can actually get the amino acids we need just rained down on us from comets and other interstellar debris. But RNA can act both as the information carrier and as a biocatalyst. If you remember last time we talked about ribosomes, and I said it was the RNA in the ribosome itself that actually catalyzes the formation of that peptide bond. You don't need any proteins on a ribosome at all to form that peptide bond. It's the RNA itself. So the RNA itself is a biocatalyst. Not only that, it has the sequence of bases that it can use as a template to make another copy of itself. So that's one of the reasons they think RNA was the original molecule that developed where all life sprang from, basically. Because you get a sequence, if it folds correctly, it can have catalytic activity, and then it can also, that sequence can serve as a template to replicate itself. This is, I guess, figure 136 from your book. They think the prebiotic formation of simple compounds led to the production of short RNA molecules with random sequences. Those random sequences would then be selected for selective replication of self-duplicating catalytic RNA segments. So segments that could self-duplicate and had catalytic activity would be the ones that would be replicated, right? Because the other ones aren't doing anything. They're just going to sit there and be degraded by other things. And eventually the RNA led to synthesis of specific peptides catalyzed by RNA. And then we see that evidence from that from the ribosomes themselves, because it's the ribozyme that actually catalyzes those peptide bonds. Increasing role for peptides in RNA replication. Assume RNA, if you get sequences that generate peptides that help protect those RNA sequences and help them replicate, that's going to be evolutionary advantage for those sequences, and they'll reproduce more than the sequences that don't use the peptides or code for the peptides. And then you get primitive translation system develops with RNA genome and RNA protein catalysis, and ultimately that will lead to genomic RNA begins to be copied into DNA. So the reason why you'll see a shift from RNA to DNA is because RNA is chemically just unstable versus DNA. That one hydroxy group missing leads for a greatly increased stability of the DNA. And that's because RNA can self-cleave itself under certain conditions. And then we end up with a DNA genome translated on RNA protein complexes, the ribosome, with RNA and protein catalysis. So complementarity in DNA allows replication with near perfect fidelity. And that's, we have two strands that form a double-stranded helix. Each strand forms or can act as a template to copy the other strand. And in the new strands, it's called semi-conservative replication, where each new strand contains one parent strand and one daughter strand. So it's semi-conservative. And I know you guys have to be familiar with this, right? The central dogma of biochemistry or molecular biology, where the DNA codes serves as the code and the genetic information. You get an RNA molecule or a messenger RNA transcribed from the DNA. That RNA then codes for a protein, and the protein sequence, the amino acid sequence in that protein, actually is going to govern how that protein is going to fold, what activity it's going to have. In this case, we're forming a hexokinase. Hexokinase is an enzyme that phosphorylates glucose or other hexoses. What do you think? So I'm jumping into terminology just to get you guys thinking about it. What do you think hexokinase stands for? So glucose. How many carbons do you think glucose has? Just it's a substrate for hexokinase. 6. So hexokinase is an enzyme that adds phosphate groups to hexoses. Hexoses are 6 carbon sugars, one of which is glucose, and that's the main substrate that we see hexokinase working on is glycolysis. So natural selection is going to favor some mutations. Mutations occur more or less randomly because DNA is just a chemical. It's going to have it's going to break down at a set rate, and mutations will be selected for if they have an advantage for that organism that has the mutation. Like you could have duplication mutations. For example, I think we as humans have duplication mutations for amylose or amylase, which is an enzyme that breaks down starches. So that's why we evolved eventually to be able to eat a starchy diet based on grains and stuff. When we started developing farms. So mutations that give organisms an advantage in a given environment will then give that organism with the mutation an advantage to be able to survive better, be more fit in that particular environment. Once we started developing farms, people who weren't able to digest all the starches weren't able to breed as much because they were sick all the time because that's all they were eating at the time. Does that make sense? So, some selections favor natural selections favor some mutations. It's the hexokinase gene. So you can get a rare mistake during the DNA replication which duplicates that hexokinase gene. A second rare mistake results in the mutation of the hexokinase gene. And you end up with, here's the original hexokinase. The mutated hexokinase then can act on not only glucose, but another sugar, galactose. And then you can use galactose. If you have this mutation, it'll be able to use galactose as a fuel source also. Yeah, also, you guys have heard of this theory, right? Where the bacteria invaded eukaryotic cells and became like symbiotically part of our cells. Meaning the mitochondria in animal cells and plant cells and chloroplast in plant cells, right? Because evidence from this is mitochondria and chloroplasts have their own genomes, their own ribosomes, and they make their own proteins. They don't get 100% of their proteins from their own genome. Some of them are imported from the main nucleus, but that's some of the evidence. It's endosymbiosis. That was it for chapter 1. We'll just rush into talk about water. Okay? We were real short last time because of that safety class. class. That I'm sure was exciting, right? So, learning goals for this are going to be what kind of interactions occur between molecules. It's like general chemistry stuff, right? So, we're going to see, if you got name the intermolecular forces can happen.
Covalent and ionic bonds.
Those are, yeah, but like intermolecular aren't like covalent bonds. The hydrogen bonds, which are a special form of dipole interaction, right? Yeah, London dispersion are like when you don't have like dipoles or not permanent dipoles. Right? That's where the molecules close together will interfere with each other's electron cloud and make like the temporary dipoles, thus forming some sort of an attraction. So why water is a good medium for life? We're going to talk about why nonpolar bodies aggregate in water, how dissolved molecules alter properties of water. You guys remember that? What was that called? Colligative properties? Remember some of those in general chem? Freezing point, what? Increase or depression?
Depression.
Yes, depression. What gets increased? Boiling point. And, oh no, we just said it, melting, or freezing point depression, what do you call it? It's directly related to the boiling point. I mean, directly related to the boiling point. No, the vapor pressure, right? You guys know the boiling point is the point at which the vapor pressure equals the atmospheric pressure. You know, that's it boils, your liquid boils. We're going to talk about how weak acids and bases behave in water. What's a weak acid? What?
There's a bunch.
Yeah, I mean, give me an example of one. Why is it considered a weak acid? We can talk about acetic acid, all right? But what is special about acetic acid that makes it a weak acid? It has that one functional group, right? Carboxylic acid, which can then donate that proton. It loses the proton. It tends to lose the proton. Why does it do that? Is because throughout that once you leave that, let that proton go, you have like a resonance between the two oxygens and the carbon delocalized, and that stabilizes the charge, which makes it acidic. And basically what I was getting at is acids give up protons. Bases tend to accept protons. And we'll see amino acids have both the carboxylic acid and acid domain and a basic domain, which makes them something called a splitter ion and physiological pH. How buffers work and why we need them, and how water participates in biochemical reactions. So, water is the medium of life. One of the reasons is we get UV protection from water. UV is known to degrade and change nucleic acids, right? So, exposed to a lot of UV. In fact, our biosafety cabinets, one way to clean them or disinfect them before you start using them, you turn on the UV light, let it sit for 10 minutes, and hopefully you killed off anything that might contaminate your samples that you're going to work in, right? Also, UV associated with skin cancer, right? That's because it mutates, forming these pyrimidine dimers in your genome. If you have too many of those in the wrong places, it's going to lead to cancer. We all contain probably 70 to 90% water. So our biochemistry happens in aqueous solutions. Chemical reactions occur in aqueous milieu. I can't even talk about that. I told you I was bad at words. Apologize. Milieu. How do you say that? It's okay to laugh at me. I'm fine. Water is a critical determinant in the structure and function of proteins, nucleic acids, and membranes. We'll see why it's important to have that aqueous environment for the proteins to fold the way they fold. If they didn't, they wouldn't fold like that at all. It's actually the water, the entropy of the water that causes those proteins to fold. Or that's the largest contributor to that free energy change. So, we all know about the structure of water. Right? You've got this octet rule, dictates that there's 4 electron pairs around an oxygen atom. They're sp3 orbitals. Two of these pairs are covalently linked to 2 hydrogen atoms. So we got this, right? So we have 2 covalent bonds of hydrogen atoms, and then you got 2 free lone pairs. And then that gives water, basically, because of the lone pairs, it's like a distorted tetrahedron. Those hydrogen are kind of pushed down a little bit. The electronegativity of the oxygen atom induces a net dipole moment, right? So we have, this is negative, this is positive. So net, you have like a net dipole where it's positive on this side and partially negative on this side, right? That makes it quite polar. And because of the dipole moment, water serves as both a hydrogen bond donor and acceptor, right? So we've got 2 hydrogens here. It's partially positive. You have another water here where it's partially negative. It's a hydrogen bond donor. And at the same time, these electrons can accept hydrogen bonds. So it's a hydrogen bond acceptor. So each single molecule can form up to 4 hydrogen bonds spontaneously. That's one of the reasons it has such high surface tension, such high energy of melting or crystallization, and so forth. So remember that 4 hydrogen bonds. There's a question on the test I asked you. If I told you last time, I don't know how many people they put 2 or 3 on there for how many hydrogen bonds can a water molecule participate in. So the strong dipole or charged dipole interactions that arises between an acid proton donor and a base proton acceptor, and typically it's 4 to 6 kilojoules per mole for bonds with neutral atoms, or 6 to 10 kilojoules for a bond with at least 1 charge. And it typically involves electronegative atoms, frequently nitrogen and oxygen. Nitrogen and oxygen, that's what we see everywhere in biochemistry. Nitrogen, amino acids, oxygen and amino acids, and water, right? And then hydrogen bonds are the strongest when the bonded molecules are oriented to maximize electrostatic interaction. Okay, so when things get colder, they get more dense, right? Right? Typically, things get more dense, they sink to the bottom. So ice is colder than liquid water. How come it doesn't sink? Any ideas? Okay, so ice is cold enough that the water's crystallized. When it's in that form, you have basically 4 hydrogen bonds occurring in the perfect water crystal lattice. Each molecule is participating in 4 hydrogen bonds. But liquid water has enough kinetic energy. The average number of hydrogen bonds that a single water molecule will have when in the liquid form is about 2.3. So the liquid water can be packed closer together because to form that crystal, there's a lot of empty space. Whereas liquid water has enough kinetic energy, you don't form the perfect crystal lattice, so you're only forming about 2.3 hydrogen bonds per water molecule, whereas the crystal, the ice formed water, there's a lot of empty space, which actually makes ice less dense than the liquid water. So that's why ice floats. And one of the reasons we're able to get life on Earth is because when the lake freezes, it doesn't kill everything in it. You get nice 32-degree water underneath for things to survive. Does that make sense? And ideally, all 3 atoms are in a line, which is what you see with the perfect crystal lattice structure of water. And this just shows you strong hydrogen bonds when things are perfectly aligned. You have the lone pair of electrons on this carbonyl here. Looks like it's part of a phosphate, not a carbonyl, sorry, but analogous to a carbonyl. Or a weaker hydrogen bond if it's not perfectly oriented. So hydrogen bonding in water, like I said, consider both as a donor and acceptor, and up to 4 hydrogen bonds per water molecule gives its high boiling point, high melting point, and its unusually large surface tension. So hydrogen bonding in water is cooperative, meaning, what is that? It's meaning when you have something donating a hydrogen bond to this, it's going to make this more likely to donate the hydrogen bond. It'll make them all kind of stronger. Right? And hydrogen bonds between neighboring molecules are weak, 20 kilojoules per mole relative to the OH covalent bonds. So just remember, these aren't strong like covalent bonds, but they're pretty good intermolecular forces there. And distances, remember, shorter bonds are stronger, covalent bonds are about less than 1/10 of a nanometer, whereas hydrogen bonds are almost 20th of a nanometer, not 20th, 2/10 of a nanometer. So ice in a solid state, I think I already talked about this. So water has many different crystal forms. The hexagonal ice is the most common, and it forms a regular lattice and thus has really low entropy because it's, crystallized. Higher the entropy, the more disordered. Hexagonal ice is highly ordered, and it contains more hydrogen bonds per water molecule. Thus, the ice has a lower density than liquid water, thus it floats. And I can't draw this good, so it's good that it's up here. But it shows you all that empty space that you get in that lattice structure. Whereas in water, as a solvent, it just, it's all over the place, and you get about an average 2.3 hydrogen bonds per water molecule. So it's a good solvent for charged and polar substances. You guys remember, if it's charged, that water will form like that shell around the charge and help kind of diffuse that charge out. So it lowers the enthalpy of that charge, basically, and spread the charge out through amongst that area. You guys remember that? The smaller the ion, basically the stronger that charge, or not stronger the charge. Charges are all equal, but the more dense that charge is, the stronger the ionic bonds you can form, because they can get close together with their opposite charges, right? So water helps spread those out. So it's good solvent for amino acids and peptides, both of which are charged and polar. Small alcohols are polar, and carbohydrates basically are hydrates of carbon. There's one carbonyl on those, and then lots of alcoholic groups on those. It's a poor solvent for nonpolar substances, nonpolar gases, aromatic moieties, and aliphatic chains. So the water it's not going to mix well with those. What water does typically is it forms a structured lattice around those substances, like aliphatic chains, which is a decrease in the entropy of the water, because it has to be highly ordered, structured around aromatic moiety or a nonpolar gas or an aliphatic hydrocarbon chain. These are some examples of nonpolar, polar, nonpolar, and amphipathic. What is amphipathic biomolecules? What do you think that? Amphipathic? Yeah, well, it's not that it can be, it's that it is polar and nonpolar. So an example of an amphipathic substance would be like I don't have it up here, but well, this is amphipathic, right? You have these actually charged groups which are highly polar, and then this is somewhat polar also, and then these aliphatic hydrocarbon chains, or these are the fatty acid chains on a diacetyl glycerol phosphate. This is choline there, I think.
So yeah, this is amphipathic. Also, this is phenylalanine. So you have this non-polar group, and then you have two, an amine group and a carboxylic acid, amino acid. Then we have a wax. Waxes are typically not soluble in water very well. And waxes are made of an ester bond between a fatty acid and a fatty alcohol. So, but you see waxes a lot on the skin of leaves and stuff. They're very impermeable to water, to protect the plants from losing water to the atmosphere. And then we see the polar groups. All of these are highly soluble in water. Glucose, like I said, there was lots of alcohols. And then this is actually a carbonyl that's been attacked by one of the alcohols. It's a form of the ring destruction. We'll talk more about that in the sugar chapter. Water also dissolves many salts. So it's got a high dielectric constant, which reduces the attraction between oppositely charged ions in a salt crystal. So almost no attraction at large distances. So water helps spread those ions out, like sodium chloride. It'll help spread the charge out, and it increases the entropy because you actually dissolve the salt, but then the water itself is going to be more organized around those ions, so there's a decrease in entropy around those ions too. So little competing things going on, and the free energy, and those are the thermodynamics of dissolving salts. Strong electrostatic interactions between the solvated ions and the water molecules lower the energy of the system, and that's because you're spreading out that charge, and so you're kind of releasing some enthalpy. The entropy, though, increases as ordered crystal lattice is dissolved, but at the same time, the entropy of the water is decreased, right? Because you have ordered water around those charges now. They're all going to be facing one direction or the other based on if it's a cation or an anion. But the fact that you get such a big entropy increase, because you've increased the disorder of the crystal lattice, taking apart that crystal lattice. This shows you that, where you get hydrated ions. And here these water molecules are going to be more ordered around the ions, but the crystals obviously, it's less ordered when you have the ions dissolved in the solution. So you increase in entropy of the salt and a decrease in entropy of the water itself. Physics of non-covalent interactions. So we have non-covalent interactions do not involve sharing electrons. Basic chemistry stuff. It's based on their physical origin. One can distinguish between. So ionic interactions are equivalent, which means that they're active charges interacting. Like, sodium is bound to chloride to make sodium chloride salt. You have electrostatic interactions between permanently charged species or between the ion and a permanent dipole. So we'll see this in protein structure. Different proteins will form salt bridges, and that gives a highly ordered shape between, let's say, an amine group on a side chain and a carboxylic acid group on a side chain. You can form positively charged and a negatively charged group, and a real strong interaction is called a salt bridge that may help govern the shape of some protein. Lots of dipole interactions, electrostatic interactions between uncharged but polar molecules. We see those in the amide bonds of a peptide bond are polar, so you get a lot of hydrogen bonding between those within a chain. That's actually what makes up the alpha helix, and you get hydrogen bonds making up the what do you call it, the beta sheet structures, or your primary structure, or secondary structure of proteins. Van der Waals interactions. So what are those? We talked about those earlier. It's where two molecules come close together and they kind of form a temporary dipole in that electron cloud around the molecule, creating an attraction between the two molecules. And then the hydrophobic effect is basically a complex phenomena associated with the ordering of water molecules around nonpolar substances. Like I said, the water forms a highly ordered structure around non-polar substance like a fatty acid or detergent, that tail, is always, you're gonna have an ordered water. So why does oil come in droplets when you mix it? Have you ever gotten like mixed oil and water and shake it up and form all these little droplets? Eventually all those droplets will convalesce into one big droplet. What's going on there? What do you think's going on when it convalesces into one big round droplet, too, round? Because all the water that's interacting with it is going to be highly ordered around it, decreasing the surface area. So when it convalesces as one big droplet, it's the water is just the system itself is trying to decrease that surface area, so there has to be less water highly ordered around it. So the smaller the surface area, the less water needs to be highly ordered around it, and the more entropy the system can have because you have more disorder in the regular water everywhere else. You don't need as much ordered water around the hydrophobic substances. And that's basically the main factor in the folding of proteins. And these are some examples they give of non-covalent interactions. So hydrogen bonds between neutral groups, carbonyl and a hydroxy group, between peptide bonds. So this is the amine group of an amino acid that's involved in a peptide bond, and a carbonyl group of an amino acid that's involved in a peptide bond. And this is what governs secondary structure formation. Ionic interactions, we'll see this on a side chain or an amino terminus of a peptide might interact with the carboxy group off the side chain or the C terminus. And then you also can have repulsion between amino groups or carboxylic acid. And then hydrophilic reactions is this water wants to decrease the surface area around these hydrophobic groups and tends to force these into close proximity. That way the water doesn't have to be around them. It just kind of decreases the surface area. And then the van der Waals interactions between these two act as an attractive force also. So the importance of hydrogen bond, it's the source of the unique properties of water. It's also important for the structure and function of proteins. You guys learned about the DNA bases before. A, T, C, and G. Why do they always paired? Do you know? No idea? It's because of hydrogen bonding between like an A and a T and a C and a G. And the structure of DNA is perfect. Those bond perfect, hydrogen bond perfect together in a double helix. So that's why you get paired. We'll see pictures of that when we get the DNA picture. They pair up perfectly. And that's one of the ways you can introduce mutations is through keto-enol shifts while the DNA is being replicated. You can get a keto-enol shift that will accidentally incorporate, let's say, a T with a C, right? And it'll shift back and you'll have a bulge and it won't, base pair properly. It'll have to be repaired. Just one of the examples of how mutations get introduced. Structure and function of DNA, structure and function of polysaccharides. So polysaccharides, we saw how many hydrogens they have, or hydroxy groups. They'll form a lot of hydrogen bonds. They make up, they're important for makeup, like cell-to-cell contact, and it's the space in between the cells. Extracellular matrix has a lot of the polysaccharides that these hydrogen bonds are important for, interacting with growth factors and producing, our extracellular matrix. They're important for binding of substrates to enzymes, binding of hormones to receptors, and they match mRNA with tRNA. Again, via base pairing, just like for copying the double helix. So these are examples of hydrogen bonds. I'm sure you guys are familiar with all of these. This is what we see in biochemistry a lot. Carbonyls in an alcohol, and nitrogen in an alcohol. It's an ether bond with an alcohol, an amine with a carbonyl, amine with an ether, and amine with this nitrogen. Anybody know what kind of nitrogen this is? What's this referred to? Schiff base. You guys heard that? And then biological relevance of hydrogen bonds, so between a hydroxyl group and an alcohol and water. We'll see a lot of that. Between a carbonyl group of a ketone and water and so forth. Between peptide groups and polypeptides. So this is the peptide bond here, and then another peptide bond here, and we see hydrogen bonding between those. It could be a beta-pleated sheet, or if it's in a helix, it forms the alpha helix. And here's what I was talking about, your A and a T. You get the perfect hydrogen bonding between thiamine and adenine. That's how, you know, why you see A's always paired with T's and C's always paired with G's. So van der Waals interactions have two components. There's an attractive force. The London dispersion force depends on the polarizability of that molecule. And a repulsive force, which is steric repulsion. You get things too close, they don't want to get that close. So, you know, they bounce off each other. And that depends on the size of the atoms. The attractive dominates at the longer distances between 0.4 and 0.7. The repulsion will dominate at very short distances. So, yeah, the closer you get, the more attraction you get, but if you get too close, it's going to repulse. Right? And then there's a minimum energy distance, the van der Waals contact distance. And these are the van der Waals radius versus covalent radiuses for bonds. You know, like hydrogen, 0.11 nanometers is your van der Waals radius versus a covalent single bond with hydrogen is 0.03. It's like a third or a quarter of that almost. Oxygen is 0.15, whereas the covalent radius is 0.066 and so forth. You get the idea. The covalent is much stronger and closer than the van der Waals. So the origin of London dispersion, so we already talked about this. You get the quantum mechanical origin, instantaneous polarization by fluctuating charge distributions around that molecule. And universal, it's always attractive. The stronger polarizable molecules, important only at a short range. And they're weak, right? Easily broken and reversible. Universal occur between any two atoms that are near each other, and the importance of the term is determined steric complementary. So if you have two saturated free fatty acids, they're going to be sterically complementary because they're similar shape. Whereas, you guys know, you heard of cis unsaturated fatty acids? You know what's special about those? You have a cis double bond somewhere, so they're not going to be able to like sterically interact with a saturated fatty acid, which is completely reduced. So you get stronger van der Waals interactions between saturated fatty acids than you would if you have unsaturated fatty acids mixed in. Does that make sense? We'll see a lot more of that in the lipid chapter. Stabilizes biological macromolecules, so you get stacking of DNA, you get the pi stacking, and it facilitates binding of polarizable light. So the hydrophobic effect, we talked about this today already, refers to the association or folding of non-polar molecules in the aqueous solution. It's one of the main factors behind protein folding, protein-protein associations, and formation of lipid micelles. Does anybody know what a lipid micelle is? I didn't think you. Don't worry. Basically, anybody know like the basic structure of a detergent? Basically, like something like SDS or just a fatty acid. Typically, you have some kind of long chain aliphatic group, and then some sort of head group. So for a fatty acid, it's a carboxylic acid. You have an oxygen here, and that carries a negative charge. So if you have a bunch of these in solution, this aliphatic tail, the hydrophobic effect of water, if you have a lot of these in solution, you'll have a lot of ordered water all around these tails. But if you have a lot of these also, these are kind of going to aggregate together into what are known as micelles, where you have the tails that interact with each other. And the charged groups on the heads, or the carboxylic acids, interact with the water. So you have all these, they form like these little spheres, basically, because you want to reduce the surface area. Plus you have something that's good at interacting with water, a charged group on the outside. And these little spheres of detergent or fat are called micelles. That's what a micelle is. Binding of steroid hormones to the receptor, it's the same thing. Steroid hormones are typically hydrophobic. They're going to bind to the receptor due to the hydrophobic effect, because the receptor portion, where the steroid binds, the binding portion is also hydrophobic. It does not arise because of some attractive direct force between the two non-polar groups. It's because of the entropy of the water is increased when these aliphatic groups or non-polar groups are forced together, decreases the surface area of water around. Does that make sense? And this is some solubility of polar and non-polar solutes. So we have the nitrogen is non-polar. It's got 0.018 grams per liter. And then we have hydrogen sulfide at the other extreme is 1,860 grams per liter, which are highly polar. So why are the non-polar molecules poorly soluble in water? It's because of this hydrophobic effect. Water has to form that structure around the non-polar groups. So low solubility of hydrophobic solutes can be explained by entropy, which I don't know, is this like the 4th time I've said this? So you must think it's important. So bulk water has little order or high entropy. Water near hydrophobic solutes is highly ordered or low entropy, and low entropy is thermodynamically unfavorable. Thus, the hydrophobic solutes have low solubility. Here's the pictures I tried to draw. It's a carboxylic acid, fatty acid again, and you form highly ordered water around that alkyl chain. So, the origin of the hydrophobic effect, consider the amphipathic lipids in water, which is this free fatty acid group is amphipathic, right? You have a hydrophobic part of the molecule and a hydrophilic part of the molecule. So the lipid molecules disperse in the solution, nonpolar tail of each lipid molecule surrounded by the ordered water. Entropy of the system is decreased. It's an unfavorable state. So you have lots of little molecules like this. This is highly unfavored. You're going to want to decrease the surface area of the highly ordered water. Like this. So now there's less highly ordered water than, around these 3 molecules than there would be around one of each of the 3, right? So you have an increase of entropy in the system. And this is just another picture showing, a hydrophobic group of molecules surrounded. That's why it always forms like a droplet shape, because that's the smallest amount of surface area that water can have. Thus, the biggest increase in entropy. This is a picture of my cell. I think mine's better. Hydrophobic groups are always going to be sequestered away from the water, and you have an ordered shell of water molecules is minimized around those. Yes?
You said this was related to detergent.
Yeah, detergents do this. So SDS, fatty acids, anything like that. This is how you clean things, because you have hydrophobic things like grease stuck in your shirt. You have a lot of micelles all around. It gives a place for that grease to go to get out of the water. kind of mixes the grease with the water, because these are like microscopic little particles that are suspended within the water because of their hydrophilic heads, and then they have the hydrophobic tails. So it's places for like the dirt and grease to mix up with, so you can clean things out together. Basically, that's what detergent's for. Anytime you have a single hydrophobic tail, it can be considered a detergent. Whereas if you have two tails or more, they don't tend to form micelles, they form bicells or bilayer membranes, which is where we get our membranes from. So hydrophobic effect favors ligand binding. So ligands often are going to bind to their binding site through the hydrophobic effect. So that's why a lot of drugs that you find tend to be, not very soluble in water. They're driven to their binding site through the hydrophobic effect. Such sites can bind hydrophobic substrates and ligands, such as steroid hormones. Many drugs are designed to take advantage of that hydrophobic effect also. And this shows you that hydrophobic effect. You have highly ordered around the substrate and highly ordered around the binding side of the enzyme. And when those two interact, these ordered molecules are gone, thus you get a slight increase in entropy based on this binding event, which is thermodynamically favorable. Does that make sense? Effects of solutes on the properties of water. So here we are at the colligative properties again, just basic old chemistry. Colligative properties include boiling point, melting point, and osmolarity. So do not depend on the nature of the solute, just the concentration, right? So it's the number of particles in that solution. Non-colligative properties include viscosity, surface tension, taste, and color, and it depends on that chemical nature of the solute. So the cytoplasm of cells are highly concentrated solutions that have a high osmotic pressure, right? You have tons of proteins and ions in solution inside the cell. It gives it a high osmotic pressure. That's why one of the ways you can lyse cells that you'll learn if you take hematology or something is just to put those cells in a water solution. You can lyse those red blood cells and somewhat save some of the white blood cells. That's through the osmotic pressure, right? You guys have heard of this. You have pure water on the outside, and then you have a solution with some concentration of solutes dissolved in water. And these two solutions are separated by a semi-permeable membrane. The only thing that can pass is the water molecules. The solute cannot. Water is going to go into this, and it'll climb up to a certain height until, atmospheric pressure is pushing down on this enough that you're not going to get any more water in. But the force it takes to push this down to the same level, that's considered your osmotic pressure. There's going to be a certain amount of force that's going to be required to do that. And that can actually be calculated. Right? You guys do that in general chem? No? I'm sure you did. Like Chem 1 or 2? Way back when? Was it like a decade ago? This can happen in the cells, right? So you have a high concentration of solutes intracellularly. If you put them in a solution that doesn't have much, water is going to flow in and eventually rupture those cells. Likewise, if you put them in a hypertonic solution, water is going to leave those cells and they're going to shrivel up. It's the effects of the osmolarity. And then we're going to see with blood, some of this osmolarity plays a part in maintaining blood volume. It's called the colloid osmotic pressure. You guys ever heard of that? That has to do with the concentration of macromolecules in the blood. We'll talk about that way in the future. So ionization of water. Here we get to the acid-base chemistry, right? We know water can ionize to form hydronium and hydroxide. So the OH bonds are polar and can dissociate heterolytically, produce protons and a hydroxide ion. So that makes water an acid and a base.
So dissociation of water is rapid and reversible process. Most water molecules remain unionized, thus pure water has very low electrical conductivity. Resistance of pure water should be 18 megaohm centimeters. Yeah. Oh, sorry. It's like out of the corner of my eye. I thought you were. So, 18 megaohm centimeters. You'll find that's what we get out of our water purifiers for the ultra pure water that you guys use in the lab as reagent water. And that's just how pure it is. So you still can get some electrical conductivity, but 18 mega ohms, that's a lot of resistance, right? The equilibrium is strong to the left, so the extent of dissociation depends on the temperature also. So protons, they don't exist just free in solution, they become hydrated, right? So they form hydronium or oxonium. They immediately hydrated to form hydronium or oxonium ions. The hydronium ion is a water molecule with a proton associated with one of the non-bonded electron pairs. So it's H3O with a positive charge. Hydronium ions are solvated by nearby water molecules, and then those protons will just shuffle around. It's not like they just stay to one water molecule, they're easily transferred to other water molecules, like this. It's called proton hopping. Here you have a hydronium, we can hop to this one, which will then allow this one to hop to that, and then we'll just go like that all the way through the solution. And you can quantitate the ionization of water. So you have water can break down to hydrogen ions and hydroxide ions, and that's going to exist in an equilibrium, right? We write equilibrium constants like this with the products divided by the reactant in this case. And the KEQ can be determined experimentally, and it is 1.8 times 10 to the minus 16 molar at 25 degrees, and can be determined from the water density. It's about 55.5 molar. So, ionic product of water is the KW, which equals the KEQ times water, which is basically the concentration of hydrogen ions times the concentration of hydroxide ions. So you have about 1 times 10 to the minus 14 molar squared ions in that solution at any time. So pure water, concentration of free hydrogen or hydroxide is 10 to the minus 7 molar, right? Or the pH, or the negative log concentration of the hydrogen ions is 7. Right? If you guys remember, pH is negative log. So the pH equals the negative log concentration of the hydrogen ion. It's defined as the negative log of the hydrogen ion concentration. So the pH and pOH must always add up to 14 in water. So the negative log of H, hydrogen ions, negative log of OH, equals 14, the neutral solution. They're both 7. pH can be negative if you have like a high concentration of acid. It'll give you a negative pH, right? So 6 molar hydrochloric acid. If you had this at 6 molar, what's the negative log of 6 molar? Or what's the negative log of 1 molar? You know? So it's 0. So 10 to the 0 is 1, right? So 6 is even less than that. That would give you a negative, negative log, right? Which makes it a negative pH. I don't know what I said made sense. But you know what I'm saying? Anything above 1 molar is going to be negative pH. And this is the scale. So you have 1 molar, pH is 0. This is a concentrated acid. And then 10 to the minus 1 molar, or 0.1 molar is pH 1. 0.001 molar is pH 2, and so forth. So this is how your pH scale works. This is some pH of some common liquids.
So gastric juice, about pH 1.5 to 1. Lemon juice is about 2, cola and vinegar about 3 to 4, red wine, beer, and so forth, coffee 5. And then we get to the basic solutions, where seawater and egg whites about 8 or so, baking soda about 9, and then it gets increasingly basic. Household ammonia is about 12, and bleach is closer to 13. And then 1 molar sodium hydroxide is going to be pH 14. So disassociation of weak electrolytes principle. So here, before we were talking about water, it's kind of similar, but we're disassociating weak electrolytes in an aqueous solution. So this example, does anybody know what acid this is? Talked about it earlier. Acetic acid. Right. So this is acetic acid. It's going to disassociate in water to form acetate and hydrogen. And then it's going to exist like this in an equilibrium, where the concentration of hydrogen ions and acetate divided by the concentration at equilibrium you get of the protonated acetic acid. And that Ka is going to be 1.74 times 10 to the minus 5. And we can solve for the concentration of hydrogen ions that we get. If the Ka is known, we can calculate the concentration based on our concentration of acetic acid and acetate in that solution. We can calculate what the pH is going to be. But some algebra is needed, but really it's not needed if the acid is weak enough, right? Do you guys remember this? So you can set up an equation. You start off with 0.1 molar for dissolving 0.1 mole of acetic acid in a liter. It's going to start off at 0.1 mole, but then a certain amount is going to dissociate to form acetate and hydrogen ions. And the Ka is going to be equal to x times x divided by 0.1 minus x. That equals, we know the Ka, 1.74 times 10 to the 5. We solve for x, we get x squared equals 1.74 times 10 to the 6 minus 1.74 times 10 to the 5x. So what's the assumption here? We can solve it the quadratic equation, and you end up with x equals 0.001310, and if we take the negative log of that, you should get 2.883. But the thing is, because this pKa, so much, this is tiny, right? Versus 0.1. Does that make sense? You see that? That's like just a little over 100th. So for a lot of weak acids, you can make this simpler, or you just make this, because what's 0.1 minus 0.0 is it's going to be 0.99 something, right? Or 989 something, basically. So it's basically negligible, the amount that gets disassociated. So in reality, you guys can ignore this x here. So you just have x squared equals 1.74 times 10 to the minus 5. You take the square root of that, it's going to be pretty close to this, close enough to where you'll get a pH that's really close to that also, which is the simplified form, right? I didn't even have to go through that, I could have just switched the slides. So the simplified form, so this is 0.00132 versus what we get before, 0.00131. So it's a tiny difference. Of course, this one's more accurate, but do we need the accuracy that good? Because it was 0.2883 versus 0.2880. So our pH meter will read to that accuracy, but it's really hard because it varies so much based on just the temperature of the solution spinning around. So this makes it easier to calculate pH. Do you think you guys can do those kinds of calculations? I think so. We can practice. So the pKa basically is a measure of acidity of something. So that acetic acid, Ka is 1.74 times 10 to the minus 5, so that gives you a pKa. Where is that? It's probably somewhere around 3.5 or 4, 4.76 pKa of acetic acid. So a lower pKa means you get more dissociation. This is carbonic acid has a pKa of 3.77. What is this? Glycine. And it's not, it's just the carboxylic acid group of glycine has a pKa of 2.34. Phosphoric acid has 3 pKa's, right? And if you look here, glycine has 2 pKa's. And so does carbonic acid. It's each protonatable, proton that can be donated, you're going to have another pKa. In this case, we have an amine group that's protonated, so the first pKa is 2.34. The next pKa, where you lose the proton from the protonated amine, is at pH 9.6, so a pKa of 9.6 here. And phosphoric acid is 2.4, 6.8, and 12.4. 3 different pKa's. So, which brings us to buffers, right? So each one of these can act as a buffer. Anybody tell me, like, where would you want to use these as buffers? Do you know? Any idea? Would you want to be around the pKa or somewhere far away from the pKa? You know? We'll get into that. So, the buffers resist change to pH, right? We knew that. And when the pH equals the pKa, there's basically a 50-50 mixture of the acid form and the base form. So at pH 6.86, half of the molecules of this phosphoric acid in solution are HPO4 2- and H2PO4. So at that pH, if you add a little bit of acid, you're going to shift. Those protons are going to immediately be sucked up by this HPO4 and convert a few of them to H2PO4. So you're not going to change the pH of the water per se too much. This is where you get your buffering capacity. Or if you add a little bit of hydroxide, you're going to take these protons rather than take the protons from the water because you're at the pH that you're at the pKa there. So that's where you get your buffering capacity, is right around where the pKa of that buffer is. Okay? Does that make sense? So, buffering capacity of an acid or anion system is greatest when the pH equals the pKa. And the buffering capacity is lost when the pH differs from the pKa by about more than 1 pH. So, in this system, if we want to, say, perform a reaction, and we want the pH of that reaction to be around pH 10, we could use either of these systems as a buffer. Right? Actually, any one of these 3. This would be the best one, bicarbonate and carbonate buffer would be the best if we wanted it to be pH 10. But it would still be, the glycine would still have buffering capacity with a pKa at 9.6. And even the ammonium would have some buffering capacity, ammonium and ammonia at pH 10. Not as much, but in this case, we want to use the bicarbonate carbonate buffer. So acetic acid as a buffer system. So what pH would we want to use acetic acid as a buffering system? Around 4.76, right? So when you're using acetic acid as a buffering system, if we add hydroxide, it's just going to strip that hydrogen ion from the acetic acid rather than the water and produce more water. You're going to be able to produce that buffering effect. And if you titrate around that pKa, you'll see that the pH starts to slow down between like 1 plus or minus 1, and you get like a flat area there where you're titrating. So the one lab I'm going to have you do, I'm going to give you a weak acid, you're going to titrate, develop the titration curve, and then estimate the pKa of that weak acid. So weak acids have different pKa's. So again, this is acetic acid, this is phosphoric acid, and then this is ammonia. So you have different buffering regions, so you have a whole host of buffers that you can use. You guys seen the Henderson-Hasselbalch equation? Do you know how to derive it? It's pretty simple. If you don't want to memorize it, it's always easy to derive. Because basically, you have a weak acid, this associates to its hydrogen ion and its anion. And we know the Ka for that acid is going to be equal to concentration of hydrogen ions divided by concentrations of the weak acids and the times the concentration of the conjugate base. You solve for concentration of hydrogen ions, then take the negative log of both sides, and you get the pH equals the pKa plus the log of the conjugate base divided by the concentration of the weak acid. Notice how this is reversed from this. That's just because this is the negative log and they just switched it to a positive log, which flips this upside down. So this makes it easy to figure out if you can't memorize it. I was always one to hate memorizing things if I could just figure them out during the test or when I need them. So, and I might give you a couple of questions where you might need Henderson-Hasselbalch. So biological buffer systems. So maintenance of intracellular pH is vital to all cells, right? So because the pH is going to basically govern how our proteins fold, and proteins and enzymes have certain pHs at which they are the most active, that are critical for metabolism. So maintenance of intracellular pH is vital. Enzyme catalyzed reactions have an optimal pH. Solubility of polar molecules depends on hydrogen bond donors and acceptors. The pH is going to change the availability of those hydrogen bond donors and acceptors, right? If you have a carbonyl or carboxylic acid, it can be a hydrogen bond acceptor, but then if it gets protonated, it can be a hydrogen bond donor, right? Likewise, an amine group can be a hydrogen bond donor, but if it gets deprotonated, it can be a hydrogen bond acceptor. Does that make sense? So pHs are going to change the protonation state of side chains in amino acids, which is going to change how they're going to interact with their substrates and how they're going to interact folding together in their structure. So equilibrium between carbon dioxide gas and dissolved bicarbonate also depends on the pH. And this is actually the buffer of our blood. Bicarbonate and carbon dioxide. And there's actually a Henderson-Hasselbalch-like equation that you can derive that instead of having bicarbonate and carbonic acid or pH, you have carbon dioxide gas and bicarbonate. And that's how we regulate our blood pH. How do we do that? Do you know? Because we control the concentration of carbon dioxide and bicarbonate. Do you know what organs do that for us?
The lungs, the carbon dioxide.
Yeah, lungs. We blow off carbon dioxide in the lungs. How about bicarbonate? Where do we get rid of that?
Kidneys.
Yeah, through your urine. It's one of the reasons it fizzes so much. It's that bicarbonate spontaneously converting to carbon dioxide fizzing out. So that's how your body controls your blood's pH. It'll shift it based on what's going on with your metabolism, either to be more acidic. If it is more acidic, it'll typically, you'll blow off carbon dioxide, shift that equilibrium to form less acid. Buffer systems in vivo are mainly based on phosphate in the millimolar range, bicarbonate, it's important for blood plasma, and histidine. This is a good buffer at neutral pH also. And then we get buffer systems in vitro that are often based on sulfonic acids, such as heaps, PEs, and CHES. And you'll see this sometimes in cell culture. But phosphate is mostly intracellular. Bicarbonate is going to be in your blood, and histidine you also find in other places. So water is a reactant in biochemistry. So
Here we have ADP plus phosphate. You remove the water and you form a phosphoanhydride, which makes ATP, and you get water as a result. And likewise, that water can be used to hydrolyze this bond and we produce ADP and phosphate. You'll see a lot of the reactions we find between molecules is a dehydration or a condensation reaction, where it's called a condensation reaction. Here we're condensing ADP with a phosphate and it's condensing the two. You know, condensation gives up, is when you get the formation of water, right? So you see the same thing, peptide bonds are formed via condensation reactions. So is the addition of DNA bases and so forth. It's all condensation reactions. So where you combine, you're polymerizing two amino acids, you're eliminating water between those bonds, basically. So water bound to proteins is essential for their function. This is an enzyme. I think you can see where all the water molecules are bound in that picture. And in this case, it's bound for water bound. It's essential for their function. This is at an active site on an enzyme. And you can see how the water interacts with all the side chains. These are side chains on amino acids. This is asparagine. 153 means it's the 153rd protein in that peptide, and it's an asparagine. This is arginine 156. And here you have water forming a hydrogen bond with the carbonyl, and then it also forms it with the amine group on an asparagine 168. And here it's at the active site where you're finding, is that glucose or fructose? That's glucose. So that's basically it for the water.
I'm not sure if it's involving the water, but you're saying something about when proteins fold. That was done through what was it again? Was it through
the main driving force for protein folding is the increase in entropy you get from the water when all the hydrophobic moieties of that protein get condensed to the core of a globular protein for the most part. So when that protein's produced, it's being spit out of the ribosome. A lot of those side chains might have hydrophobic side chains. As that protein folds, those hydrophobic side chains get buried to the core of the protein. And it does that, and this whole class of proteins that are known as globular proteins have a lot of hydrophobic moieties that get buried to the core, and it's that they're globular because they're like little globules of oil. They're like spherically shaped, and that decreases the surface area of the water that has to hydrate those hydrophobic moieties. So you get a big increase in the entropy or disorder of the water because there's less that has to be ordered around those.
the protein is made with that in mind, knowing that it'll fold,
Yeah, it doesn't necessarily know it, but evolutionarily, those sequence of amino acids fold that way,
and
it's driven by the hydrophobic effect for the most part. But then you also have things such as salt bridges that can form at the core, and your primary or secondary structure where you're forming alpha helices. Because inside the core of that globular protein, you still have polar groups that form the peptide bond, that amide peptide bond. You have a nitrogen and a carbonyl there. Those hydrogen bond with each other, which helps occupy that, the polar regions within the buried chain. We'll talk about that more in the peptide chapter. Just know that water plays an important role in how it's going to fold. And this is helping to stabilize, hydrogen bonds with the OH groups on a glucose at the binding pocket of the enzyme.
So those waters stay there, pretty much.
Yeah. Some waters are like permanently part of a protein, but then sometimes they're not permanently there, but they can be replaced by other water molecules. But yeah, there's water molecules there, depending on the kinetic energy.
there's an energy barrier for them to yield, to leave, because you gotta break these hydrogen bonds. So kinetic energy can do that, but then there's thermodynamically, you'll get a release of enthalpy if another one comes and binds to the exact same place, right? So again,